last week was basically with slide 22 with mutation, and I said, hey, there's a video link here. It would be really great to watch that, or and I actually took this video link and I put it in the list in Canvas for you. And this just is a really nice summary slide that says, hey, there are tons of different ways environmentally for your DNA to get messed up during your lifetime, and then your cells have to repair it, right? So that's the end of the first half of chapter 9, which deals specifically with replication of DNA. And remember, DNA is replicated prior to mitosis and meiosis. Well, what's happening during the rest of the cell cycle during G1 and G2 when the cell is taking care of normal business? It needs proteins, right? So transcription and translation are the process by which DNA gets copied in order to make workable functioning proteins for the cell. And so in order to copy the genes from DNA that code for proteins, we need 3 types of RNA, excuse me, to get that job done. And so those 3 types are messenger or mRNA, transfer or tRNA, and ribosomal or rRNA. And they are all involved in manufacturing polypeptides. And all 3 types of RNA are made in the nucleus. The focus in the textbook is on messenger RNA, and tRNA and rRNA get mentioned just a little bit, but they're all a part of the complex that is used to manufacture polypeptides. And if you look at this slide, which is slide 24 in the PowerPoint, it gives you some more details about the 3 types of RNA. So messenger RNA, mRNA, is a single-stranded nucleic acid that is made from either one or the other strands of DNA. And both strands of DNA and the double helix code for proteins, but only one section, one side is read at a time when the DNA is unzipped so that you can transcribe RNA. And ribosomes, they're non-organelle cellular structures. We don't strictly call them organelles because we like to say, hey, only eukaryotes have organelles. And ribosomes are these molecular complexes that are found in both prokaryotes and eukaryotes. So out of convenience, we say, okay, ribosomes, because they're this conglomerate, they're not really polymers, and they're not really organelles, and therefore it's okay for them to be both in prokaryotes and eukaryotes. See how we get around that organelle thing? Mm-hmm. We just say they're not. And so a ribosome is protein and ribosomal RNA, so special type of RNA that bonds with proteins to form structures we call ribosomes. And a functioning ribosome has 2 parts. It has this large subunit. I got to remember how to do the little drawing on it. It's just the PowerPoint isn't responding. That's all right. I was going to get my little scribbler out, see if it will let me. Oh, be nice. Sorry, I'm having trouble remembering how I did that before. Insert?
Yeah.
Okay, I'm in insert, but then
it's shapes.
It is shapes. Okay.
Yeah.
Oh, because it's the squiggle. It's this guy.
Yeah.
All right. My brain just froze there, kind of like my computer, at least not for 10 minutes. All right. So I just wanted to highlight that there are these 2 parts here. There's the large subunit, and I have to figure out what it is about this that is making it go dark.
Yeah, you have to find the thing that says clear. I forgot what it's called. Oh, it's in the tools.
Yeah, but it shouldn't be filling it. It shouldn't be filling it in, like just to draw a shape, right?
It does.
Let's see. If I say fill, and I say no fill, I wonder if it will stay that way.
Yeah, it will.
Well, that will, but I mean if I go and grab it the next time.
Oh, that would be nice.
Because it should just do that. So here's the large subunit, here's the small subunit, okay? And it's gonna fill it again. Maybe I just need to not make a full circle, and then it won't, and then it won't fill it in.
Yeah, I think that works.
Okay.
Yeah.
All right, so we'll try that. All right, so when each of these two parts is part of the whole ribosome, and the ribosome again is the rRNA combined with the protein, and in this large subunit we end up with actually two sites that are binding sites for tRNA, for transfer RNA to come on in and combine temporarily with the ribosome complex. And you know, basically what happens is the subunits of the ribosomes bind with the messenger RNA, and then that creates these slots for the tRNA that become active, and then the tRNA can come right here, and it has this exposed section called the anticodon, and the anticodon is going to be a 3-base sequence that's going to be exactly opposite to a certain codon on mRNA, and because they are opposite and therefore complementary, they can hydrogen bond together, and so 2 tRNAs at a time can be in, you know, these 2 sites, one in each of them, and they drop off an amino acid. So these 3 types of RNA are all involved in the process of translation. So transcription is how all the RNA is created. Translation is how they all perform a function to synthesize a polypeptide chain.
Sorry, can you say that one more time?
So transcription, oh gosh, I sure hope I didn't say it backwards the first time.
Oh no.
It's like mitosis and meiosis. Who made those 2 M words that were so similar, just so that like even in their grave they could be laughing forever after? So transcription is first. So transcription is how we create a working copy of a segment of DNA, and translation is how we use the mRNA and the tRNA and the ribosomes with their rRNA working together to create a polypeptide. So we transcribe DNA to RNA, and we translate RNA to a polypeptide. Transcription and translation.
I feel like one of those videos said the C in transcription comes before the L in translation.
Oh, to help you remember what comes first?
Yeah, so transcription comes first. Is that
Well,
just the opposite of what you just said?
No, transcription and then translation. So transcription and transcribe. Translation is, so when you transcribe something, you're performing the process of transcription. And when you translate something, you're performing the process of translation. So here we have a really, really short segment of DNA that you can see as anti-parallel. And one of these strands we're going to use to make a polypeptide. And so looking at these, we're talking about, okay, complementary or coding strand and template strand. Which strand of DNA gets called template and which strand gets called complementary or coding really depends on which one is being read to make the mRNA. And so the template strand, the one that we named the template strand, is the one that the cell has directions to have be read and to make mRNA. And the complementary or coding strand actually is going to match the mRNA. It's going to be opposite to the template strand. But think about this. When we make mRNA, we come along and we go along the template strand, and the enzyme that is doing that process that is transcribing the DNA to RNA is going to read the base sequence of the template DNA, and then it is going to create a strand of RNA opposite and therefore complementary. And so the RNA that is made is going to be complementary to the template strand, just like the complementary DNA strand is opposite to the template. And then if you look at mRNA versus tRNA, what do you notice about their codons?
They're complementary.
They're complementary also. And that means that a 3-base codon can match up with a complementary 3-base anticodon. And so that will tell you that for a 3-base sequence of mRNA, which is what we call a codon is a 3-base nucleotide sequence that codes for a specific amino acid, okay? And the tRNA that is complementary to that 3-base sequence is the tRNA molecule that will be permitted to bind temporarily opposite and drop off its amino acid. So there is only one codon for methionine. It happens to be the start codon. And in mRNA, that 3-base sequence is AUG, and the tRNA is UAC. And so only a tRNA that is UAC can carry methionine and come and drop it off as the initial starting polypeptide. So the first amino acid in every polypeptide chain is always methionine. And it's because the start codon for mRNA is AUG, and it codes for methionine. There's only one start codon. And then after that, everything that is downstream is what we call it. Everything that comes after the 3-base sequence that's AUG, we would mark as a codon. So the next 3 in the sequence code for a different amino acid and so on. And so if you had a chain that was like 2,000 codons long and you took one of them out and it was a deletion, what would happen to the codon reading all the way downstream? It would all shift one spot. And so then every codon would change. So what would happen to all the amino acids in that polypeptide sequence?
They would change.
They would change. And so an insertion that adds one, I almost said amino acid, an insertion, a mutation in DNA that adds one base and leads to a change in codons in RNA, or that deletes one base, usually leads to a whole completely different protein. A swapping where you just have like one nucleotide base up here changed for another might change a single amino acid and not the whole function of the protein, unless it accidentally changes it to a stop codon, and then the protein isn't complete. And you look at that in I think it was the learning assignment last week on mutations. Does that sound familiar?
Yeah.
Where you went over that a bit? Okay. So this is a visual diagram that reminds you that in eukaryotes, transcription, making the expendable copy of DNA, making the RNA, transcription happens in the nucleus. And whether it's mRNA or tRNA or rRNA, they're all made here. And then they are sent out either to the rough ER or into the cytoplasm. And then translation is when mRNA and tRNA and rRNA all combine together with this ribosomal complex to make a protein, which is a functional, folded active polypeptide. So the slide that was in between just reminds you, hey, it's 2-step to make a protein, and remind you of what I was saying about transcription and translation. Okay, there are a couple of videos about transcription and translation, and there's also one done by the Amoeba sisters about how to read a codon table. And if you're struggling with this at all, that might be a good video to watch. But one of the questions, it's either in the lab or it's in the learning assignment, I'm not sure which place, asks about the redundancy of the codon table. Anybody remember how many amino acids there are?
60.
20.
There's 20 amino acids, but do you know how many codons there are in this table?
61.
There's, well, technically though, 64.
Okay.
To see, we have 4, 8, 12, 16 by 16 by 16 by 16, but 3 of them happen to code for stop.
Okay.
But there's 64 codons, 61 of them code for an amino acid, and 3 of them code for stop. Well, how do I go from 61, right, to 20? Well, if you look at this right here, how many codons for leucine? 6, right? 6 codons code for leucine. And so that means that there's a little bit of wiggle room here. They all have U in the middle, but there's a little bit of wiggle room. So if there's a mutation in the DNA sequence, is there a pretty good chance that you might still get a leucine out of it anyway?
Yeah. In the lab on page 7, there's a table to fill in. And in one of the columns, the only information we have is that the amino acid leucine is produced.
So you work backwards and you can end up with any one of these.
Okay.
It doesn't matter which one you do.
Okay, while you were just talking, I was writing down like the 6 possible
You
codes.
could fill it in with all of them, but you could just choose one. My key has all of them, but yours can just be one.
Okay.
And so what it means to be redundant is that there are multiple codons that code for the same amino acid, and that protects the cell from the negative consequences of mutation. So you can have some mutation that doesn't actually necessarily result in a different amino acid being brought in to the polypeptide chain. And it kind of makes sense evolutionarily that there would still only be one start codon, because you don't want to accidentally mutate into start. And so if there were multiple start codons, it would be pretty easy to have shorter mutated, fragments of a protein, but there's only one. And so the lack of redundancy there ensures that proteins tend to start where they're supposed to. And the fact that there are 3 stop codons that are very similar is a good help to make sure that a mutation in the stop codon doesn't accidentally result in something other than a stop. The protein still gets stopped where it's supposed to be. Now, is it fail-safe? No. But there are a lot of mechanisms that the cell has to proofread DNA when it's being copied, and to proofread mRNA, and to make sure that only the tRNA with the appropriate anticodon that's complementary to the RNA codon can bind. So there's a lot of steps that the cell has in place that protect it from the negative consequences of mutation during that cell and that organism's life. And so remember, this is all dealing with cells that are part of a eukaryote that are manufacturing protein for that organism to function. It doesn't have anything to do with reproduction. It's all about the work and the life of that cell right now when we're talking about transcription and translation. Okay, so remember that we see DNA and RNA in all living organisms. And viruses are not considered living, but viruses either have DNA or they have RNA. The current coronavirus that we're dealing with is actually one that has RNA to keep its genetic code. It doesn't have DNA at all. And that can be, so there are viruses that have RNA only. And then basically what they do is hijack the protein manufacturing and the RNA synthesis of a cell. They don't need to get into the nucleus. They just need to take over, you know, the processes of transcription and translation so that they can get the cell to make all the proteins that are necessary to assemble to, you know, new virus molecules. And it's not like a virus has a brain and is doing this intentionally. Please keep that in mind. It's just that once the molecule is pulled into the cell by the cell, it becomes a part of, you know, the manufacturing process. It's like if somebody just put a code in for a new kind of you know, cake to be made at the Little Debbie plant, it would just become a part of the process in there that was done automatically. Does that make sense?
Mm-hmm.
Why I thought of that, I have no idea. Oh, I know why it is, and this is hilarious. It's because one of my daughter's assignments was to watch a video about all the different components at a recycling plant. So that must be why I thought about it. So in both transcription and translation, we see these terms initiation, elongation, and termination. And they mean exactly that. Initiation means to start the process, elongation means to continue it, and termination means
Stop.
stop, right? So when you look at a eukaryotic chromosome, any one of, you know, a human's 46 chromosomes, it's 23 pairs. If we look at chromosome 2 doesn't code for just one gene. It has many genes. And so there will be a section of the DNA right before the gene that's called the promoter. And that is the place that the enzyme RNA polymerase can actually bind to signal the DNA molecule to unwind at that point, and therefore to open for the length of the gene. So this promoter is a little nucleotide sequence that is before the gene code itself. And it's basically an attachment site for a specific enzyme, RNA polymerase. And so initiation is the step of the RNA polymerase binding to the promoter. And then elongation is when the polymerase goes along that template strand of DNA and creates the complementary strand of RNA, the one that matches, okay? And then termination is going to be when it gets to, you know, the end. And basically, the next slide says transcription stops when the RNA polymerase reaches a termination signal. So there's another nucleotide sequence at the end of any gene that says, oh, that's it. And so then the RNA polymerase is decoupled. It separates from the DNA, and the hydrogen bonds between the complementary strands reform. And so this is what it looks like when we are talking about initiation, where it looks like this big claw, the RNA polymerase, and it just shows that, hey, there's this section on the DNA for whatever gene the cell has given the signal that needs to be read to make a particular protein. And we could get into molecular signals and how all that works, but not in Biology 160. We just jump straight into what are transcription and translation for now. So this RNA polymerase has been given the signal to go bind and transcribe gene number 3 on this particular DNA molecule. And so the polymerase binds to the promoter, that length of DNA that says, hey, over here, this is where the gene is, and that we call initiation, okay? Elongation is the polymerase moving down that open segment of DNA that codes for the protein, moving along the template strand, and one by one, pulling in free nucleotides that are in the nucleus that have been manufactured and putting them in the order dictated by the sequence of bases in the template strand. So if you remember, we have this opposite strand that's not being read. We call it the complementary or coding strand, because not only is it complementary to the DNA on the other side, but it's also coding or matching exactly the RNA, except for everywhere there is a T on this strand that's not being read, there is going to be a U in the RNA. So that's why you sometimes see this strand that's not being read called the coding strand. So this is elongation, and elongation continues until we get to what point? Till we get to the termination signal. And it's just basically a little length of DNA that says, that's it, folks. And we call it the termination signal. And the RNA polymerase lets go, and the DNA helix re-hydrogen bonds. And then what do we have? We now have RNA, okay? And that RNA is now an expendable copy, something that can leave the nucleus, and the DNA is protected, and it can be sent to where it needs to go, and it can go be part of the protein making process, okay? So transcription is how we get an expendable copy of a length of DNA that codes for protein. And then what do we do next? Well, we don't have a protein yet, so we gotta go make one, okay? So one of the things we haven't gone over yet is, guess what?